Hello, my name is Rania, and in this video I'll explain hemoglobin and oxygen binding. So what is hemoglobin? Hemoglobin is a 4-subunit protein that is a component of red blood cells. It also has an oxygen binding pigment called heme, and that is what gives blood its color. It efficiently carries oxygen from the lungs to the tissues and contributes it to the transport of carbon dioxide and hydrogen ions back to the lungs. So let's take a closer look at heme. Hemoglobin depends on the presence of a bound prosthetic group called heme, so that hemoglobin can bind to oxygen. It consists of a central iron atom and an organic component called protoporphyrin. And protoporphyrin is made up of 4 pyrrole rings linked by methine bridges to form a tetrapyrrole ring. So here in this picture of the heme structure, we have 4 pyrrole rings, 1, 2, 3, and 4. 4 methyl groups, so here are methyls, and 1 right here. 2 vinyl groups, 1 and 2. And finally, we have 2 groups of propionate side chains. And how does oxygen bind to heme? So as we have discussed in the previous slide, that the iron atom lies in the center of the protoporphyrin bonded to the 4 pyrrole nitrogen atoms, and the iron ion can form 2 additional bonding sites, which are called the 5th and 6th coordination sites. So the 5th coordination site is already occupied by a proximal histidine residue, and the 6th coordination site in deoxyhemoglobin remains unoccupied and it's available for oxygen binding. And under normal condition, the iron is in the ferrous oxidation state, and it sits about 0.4 angstrom from the porphyrin plane, and as we can see here that it's too large to fit in the plane. Therefore, it's harder for it to fit into the well-defined hole within the porphyrin ring. And we also have a second histidine called the distal histidine, which is at the opposite side of the heme. So this is the distal histidine, and it prevents the oxidation of the heme iron to the ferric ion, which cannot bind oxygen, and also reduces the ability of carbon monoxide to bind to the heme. So when the oxygen molecule binds at the 6th coordination site, the iron ion rearranges the electrons within the iron so that the ion becomes smaller, allowing it to move into the plane of the porphyrin, and the bound oxygen is stabilized by forming a hydrogen bond with the distal histidine. And I also want to mention that oxyhemoglobin is in the R state, which is relaxed, and the deoxyhemoglobin is in the T state, which is less biochemically active and has a lower affinity for oxygen than does the R state. So the structure is said to transition from T state to R state. So we move into the binding capacity of oxygen to heme in the lungs. So the partial pressure of gases, temperature, and pH all affect the binding capacity of hemoglobin. So in the lungs, high pressure of oxygen along with low temperature and low pH aid in the binding of oxygen to hemoglobin. The oxygen affinity of hemoglobin decreases as pH decreases from the value of 7.4 found in the lungs at 100 torr of oxygen partial pressure here in this graph. So we have the pH of 7.4, which is the red curve, and it decreases to the pH of 7.2 and an oxygen partial pressure of 20 torr found at the active muscle. And this difference of pH and partial pressure results in a release of oxygen from oxyhemoglobin. Also, a decrease in pH shifts the standard curve towards the right, and this occurs because a greater hydrogen ion concentration, various amino acid residues such as histidine, stabilize deoxyhemoglobin in the T state. The T state has a lower affinity for oxygen than the R state, as I already have mentioned. So with increased acidity, the hemoglobin binds less oxygen for a given partial pressure and more hydrogen ion, and this is explained by the Bohr effect. The binding capacity of oxygen to heme in the capillaries. So when it moves into the capillaries, there is low oxygen partial pressure, high temperature, and high pH, so completely opposite condition of the lungs, which causes a release of oxygen when it converts into CO2. How can CO2 interacting with water affect the pH of the capillaries? So carbon dioxide in the tissues diffuse into red blood cells. And inside a red blood cell, carbon dioxide reacts with water to form carbonic acid in a reaction catalyzed by the enzyme carbonic anhydrase. Then carbonic acid dissociates into HCO3 minus and H plus, resulting in a drop of pH inside the red blood cell, causing hemoglobin to release more oxygen. So most carbon dioxide is transported to the lungs in the form of HCO3 minus and then released into the blood plasma. And this concludes my video. Thank you.